So is everybody good? Do you have anything to add, Omar? Do you think we should just get into it? It's
Yeah, sounds good to me. Sorry, I couldn't find my unmute button. We have, I think people are still joining, so
yeah, it looks like.
and we are right after the hour. I think we should just go ahead and get started because we're starting with a general introduction, right? You're doing an introduction on the
Right.
expression data,
And it'll
so
be
recorded. Yeah.
recorded, yeah. So okay. Hopefully I won't go longer than 10 minutes. We'll see. Okay. Need to adjust some things. Okay, so just 4 or 5 slides about the process of RNA sequencing and other stuff, right? So here's a little picture of the central dogma, right? DNA goes to RNA, goes to protein. And we as biologists or biochemists are interested in these processes. So to measure DNA from an omics perspective, we have DNA sequencing. If we're interested in transcription, we can measure that with RNA sequencing and microarray. If we're interested in the process of translation, we can measure active translation by riboprofiling. We have some of those experiments in some of our sites. And if we're interested in whether a gene is expressed at the protein level, we have proteomics. And that data comes to us in a little bit different forms. We'll talk about that. So I'd like to go over RNA sequencing a little bit. For RNA sequencing, we download the raw data from the repositories and reanalyze that data. And the reason we do that is we want to make the data comparable across all data sets. So no matter whether your data was collected in 2002 or 2022, it's all analyzed by the same modern bioinformatic techniques, and we can compare them on our site in our systems. The numbers produced will be different from what you see in the publication, but the trends in expression value between genes or over time will be the same. And we QA that pretty heavily. So we're confident that we're providing good, or we try very hard to provide a good analysis. So here's a little cartoon of how we analyze RNA sequence, right? We start with the raw data, and we do some quality control on that with Trim-O-Matic. Then we align the reads to a reference genome. And from this, we can determine the transcript structure and abundance. And eventually, we end up with a table of numbers that we use on our sites. And if the experiments were done with biological replicates, so 3 controls and 3 samples, for instance, then we can actually also send that through a statistical analysis program and offer data based on confidence levels. So, and from our tables of expression values per sample, we create 3 data mining avenues, right? We have the strategy system. Sorry, I'm pointing at the wrong thing. The strategy system for genome-wide searches across data types. So you can analyze from a genome-wide perspective. And we integrate each gene's expression on the gene page, so you can dig deeper into a single gene or feature. And there are graphs and tables on each gene's gene page in the transcriptomics section. And then we plot the alignments against the reference genome for visualization purposes. And with this, you can compare different data types stacked on each other in the genome browser. And we've experienced, I think, all of these yesterday, looking at gene pages, did a little bit of genome-wide searching, and looked at some alignments. Okay, microarray analysis is another technique for analyzing the transcriptome, and we have a lot of this data on our sites. In general, a microarray is a glass slide with cDNA called probes that are stuck to the slide, and the cDNA represents the whole genome. So to measure the abundance of transcripts in your RNA sample, then you convert that to cDNA and incubate that with the array. And then you use a fluorescence protocol to produce fluorescence where any of the cDNA in your sample actually hybridizes to a probe on the slide. And from that, you can tell the abundance of a transcript in your sample. So there's essentially two types of microarrays that we have data from on our site. The first is a glass slide microarray, and the microarray is sequenced in a lab and then spotted actually onto a glass slide. And this offers a two-channel data collection protocol, so you can compare two samples on the same slide. Oligonucleotide arrays, the cDNA is actually synthesized on this slide. It's a single channel setup. So if you want to determine relative abundance, you'll need to, the experiments are done with an internal standard. And again, we get data in the form of a table for that. Okay, a little bit about proteomic analysis. Well, first, proteomics, the proteins are much more complex than DNA and RNA. There are 21 or so amino acids, but only 4 nucleic acids, right? So precipitating and solubilizing proteins is much more difficult from a biochemical standpoint. You cannot be certain to isolate every protein from a mixture because the chemical properties of the proteins vary so much. But at any rate, proteomics is still very useful. We have a lot of that data on our sites. And to produce this data, you start with a protein of your sample, say trophozoites from your organism. And this is present as a protein mixture. Now, to make that amenable to laboratory techniques, you will digest that into peptides. So you have a peptide mixture. And then you separate those peptides on an HPLC and measure their mass with a mass spec. The data that you get from that is like a fingerprint of the ions within that protein, that peptide, and its molecular weight. And from using that data, then you search a database and come up with the identity of the peptides in your protein sample. And from that data, you can back calculate and map those to genes and find out which genes in your sample had peptides in your sample. So the data comes to us in two forms, right? We get tables of peptide data. Depending on how you set up your experiment, for example, you might in vitro label your cells with isotopic compounds. You might only get numbers, abundance values of your peptides, not peptides. In some cases, we get both peptides and abundance values. So with that, I'll turn to the site. And let's take a quick look at the searches, right? So let's talk about the transcriptomics searches first. I'll go to searches, genes based on transcriptomics, and we have 3 types here: microarray, RNA-seq, and single cell. I'm going to choose RNA-seq evidence. And from there, I go to this massive list of all of the data sets, RNA-seq data sets that we've integrated. In the first column, we have the organism, the genome that this data is aligned to. In the second, we have a data set the data set name, and each of these is actually a link to the data set page, which has all of pertinent information about that data set. At the top, there's a summary, a link to the publication, all the searches and that are available for that, and then an example of what the data looks like. I'm going to go back to the RNA-seq page. So in the third column here, we have all of this. We have links to all of the searches that are available for that data set. And there's three major types, right? For this, I'm going to there's differential expression, and that's when we get data that we were able to do statistics on. We have fold change, where we sort of ignore the biological replicates and allow you to analyze in a very, very useful way. And then percentile searches. Let's look at the differential expression. Okay, and I'm on the differential expression search page for these gametocyte RNA data set. And I have several different parameters that I need to tell the query before it can return me genes. I choose the experiment, and these are always set to the sense strand. In very kind of rare cases, some people will want to search the antisense strand, but I choose the sense strand, and then I choose the two samples that I want to compare. Say I want to compare male gametocytes to female gametocytes. And I can choose to look for genes that are downregulated in male versus female, that are upregulated in male versus female, or both. Like, we don't care about the direction of the change. I can choose the intensity of the change and then a p-value. So anything, I'm going to put 0.01. So anything less than 0.01 p-value will not be returned by the search. And there is our answer. We found 1,442 genes that were differentially expressed between male and female gametocytes. And here's my results. Let's go back. So in the differential expression search, right, you can only compare two samples. Well, in this fold change search, it's organized a little bit differently. You still have, you can choose the direction of the change, the intensity, but here you can group samples a little bit. Like, I can say I want to compare male and female gametocytes compared to erythrocytes, which was not an option in the differential expression. And with that, I can get answer. And in general, you'll end up with more hits from a fold change search, just because you're not ruling things out based on statistical averages. So you have to be a little bit careful about that. And we'll talk one more about the percentile search. So in the percentile search, we take our table of genes in one column and expression values per sample. In this case, we have data for 3 different samples: female gametocytes, male, and erythrocytes. And for each of those samples, we sort the data from low expression to high expression, and then apply a percentile rank to each of those. So and then we create this search where you can say, show me all genes that are expressed in the top 90 to 100% of values within that sample. So it's pretty useful. There are some caveats, as there is in anything in science. But here we were able to find 518 genes that were in the top 10 percentile of their expressed genes in that sample. Okay. And so, we have similar searches set up for microarray evidence. This DC search, direct comparison, is the equivalent of the differential expression, and then we also have the fold change and percentile. In some cases, we have specialized searches depending on the data set and how that was collected and analyzed. And in proteomics, let's look at that. We have 3 searches. Oops. The mass spec search, which only has 1 parameter. Oh, well, you choose a sample. Clear all. You choose an experiment and a sample. So let's say I'm going to look at this nuclear proteome. I'm going to say return me genes that have at least 1 mapped peptide, each aligned to that peptide, and I can click get answer. So a genome-wide search based on mapped peptides. And the quantitative mass spec is similar to our differential expression or fold change, right, from the RNA-seq. That's about it. So our hands-on exercise will go through several of those different types